It is a great honor today for me to present a special lectures, the EDAB lectures on neuroethics in the FENS forum. Today, I would like to talk about the non-human primate research for neurobiology and biomedicine. Along the way, I would mention the topics of neuroethics that are relevant to this research. Now, for people who are working in non-human primates, the goals are very clear. Most of us like to study the neural basis of higher cognitive function that are unique to the primate species, to the humans in particular. Second, we would like to understand the evolution origin of the human intelligence by comparing the cognitive functions in humans and related non-human primate species. Thirdly, we hope to generate models of human brain disease disorders, and by generating monkeys by genetic means or by treatments, so that the monkey would have symptoms of human disease for studying the drug development studies or other treatment studies as a preclinical model animals. Now, for all these studies, we clearly have to establish a rigorous ethical practice because this is the fundamental and is particularly relevant for the non-human primate research. We also hope to communicate the importance of non-human primate research to the public so that we can receive societal support of our research. In order to illustrate these three aspects of non-human primate research, I will first present some examples from works from our laboratories in Shanghai and other places in China. Now, first, I would like to present a case of a study of mirror self-recognition. Now, first, I would present a film made by a clinician on AD patients. This old lady went to the supermarket with her son and start talking to the mirror, and apparently having a very intelligent conversation. It is clear that this lady cannot recognize herself in the mirror and thought she was talking with another lady. Now, mirror self-recognition has been used as an indication of self-awareness or body consciousness. Human babies usually acquire the ability by 2 years of age. And some great apes, but not monkeys, are capable of doing this mirror self-recognition. Now, there's also observation that psychiatric or autistic patients are impaired in this ability of mirror self-recognition. And it's not clear whether it's due to deficiency of self-awareness or due to other aspects, such as loss of interest on themselves. So it would be very nice to have a model system, a monkey model, for example, that acquire the ability of mirror self-recognition, and we can study what are the neural bases for the emergence of this ability. Now, a few years ago, my colleagues have started first using a training method. I have a hat-fixed monkey in front of a mirror, in front of a mirror, laser light with higher power that's shining on the face that produce a warm feeling. The monkey are trained to touch the spot on the face by seeing the spot in the mirror. So this established this visual somatosensory association. What I show you is at the late stage of training, which took a few weeks. And the monkey, after this training, most of them can do mirror self-recognition, apparently recognize themselves. Now this approach was somewhat criticized because we are training it to touch the face. And the mirror self-recognition test is a face mark test. So we later developed another approach, which without touching the face, monkey is supposed to touch a spot on the back of the that they cannot see, and the laser spot they need to touch the spot in order to receive the reward. Now this took many weeks of training. Initially they cannot do it, but finally they can do the coordination of their own body based on the image in the mirror. So this is a proprioceptive sensation, its relationship with the mirror image. Right? This association, once established, they can do various tests of mirror self-recognition. One test is to do face mark test. Put a mark on the face, the monkey will touch the mark on the face. Or putting it on the ear, they would touch the ear. Now we found that 5 out of 7 monkey trained acquire this ability of mirror self-recognition. 2 of them fail to do this consistently. So most monkey can learn. Now this is an example of one monkey have learned face recognition, touching the face. The mark was put on without the monkey's awareness. They would do this by seeing the face image. And more convincingly is that there's a spontaneous behavior. For example, pulling the hair on in front of a mirror. Now this was not trained, but they do this. They also would look at the places they couldn't see before by looking at the mirror image. Now the other monkey with the yellow collar was not trained. He could not learn to do this by co-housed for half a year. Right? So the training is necessary to establish the association. Now, what do we find from this model? First, a monkey could learn mirror self-recognition that could be used to study neural mechanism underlying the self-awareness. So we're now putting monkey in the brain imaging machine, train the monkey before and after training, and what happened to their brain activity after they see in the mirror, in the machine, seeing themselves in the mirror, and look at the activity in the brain. And this is a way to study how the self-recognition appear, what neural basis would be. Now, this study also tells us that the mirror self-recognition is probably only an experimental tool to reveal self-awareness. Because the monkey, by doing this association, they learn the use of the mirror to reflect themselves. This ability, the lack of this mirror recognition does not necessarily mean the absence of self-awareness. This happened to be the view in the past 40 years. People use this mirror test for the sign of self-awareness. We don't think this is a good criteria. Many animals could have self-awareness, and they just have to know what the mirror what image in the mirror actually reflects. So the impaired ability of mirror self-recognition found in patients may indicate a defective association, visual and somatosensory or proprioceptive association. This one might think of a therapy in terms of the training, similar to we have done in the monkeys. Now, human babies acquire the ability by 2 years of age. Is this trained, or is this learned, or is it spontaneous? Now, we think that these 2 years has happened at the time the babies start to be aware of the meaning, and they are probably learned by the parents hint that the mirror, the image in the mirror is the baby, and gradually learn the mirror and self-association. The other approach to studying the monkey as a model is to do genetic manipulation. Now, there's now a very efficient way of manipulate genes in the monkey, in rhesus monkey, macaque monkey, such as cynomolgus, which is the most commonly used species. Now, the way to do it is to introduce a gene, human gene, by an antivirus infection or by CRISPR-Cas9 approach, editing the gene in the in vitro fertilized embryo. And this, one can do this in the oocyte and then do in vitro fertilization, or do it after the in vitro fertilization. The embryo developed can be implanted in the surrogate mother to obtain the baby. Now, one experiment was done in Kunming by Dr. Su Bin and colleagues recently by putting human MCPH1 gene into the monkey, the rhesus monkey. In this experiment, they thought this gene can control the development of the brain. It's known that the deficiency or mutation of the MCPH1 gene caused microencephalon. Microencephalon, a well-recognized brain development disease. Now, what they found is that by expressing human gene in the monkey, they found that the neurons and the cortical structure had developed slower. The delayed maturation eventually reached the normal level, but the development is slow. And very similar to the developmental delay or neoteny in the brain, in the human development. There's also looking at the genes, expression of neuronal differentiation gene or synaptic signaling gene also show a delayed expression. Now, more interestingly, they took the monkey, which is a few years old now, to perform the short-term memory test, the delayed match to sample task, and show that they are better. They have a better memory than the control. Now, this experiment has created a lot of media interest. Whether it's ethical to put human gene to the monkey brain just to understand the neurobiology of the human brain development. Now, this is something debatable. We can come back to this issue. And I think this is addressing a very important issue of the neoteny of the human brain. What is the genetic basis? And this is a frontier research that needs to be considered. And I think we can discuss whether there's ethical consensus on this type of research in the future. Now, the other experiment is to put in genes for disease, creating disease model. A few years ago, my colleagues here in Shanghai have put a MECP2 gene into the cynomolgus monkey by using lentivirus expression and a human synapsin promoter to drive a human gene, human MECP2, overexpress this gene in the monkey. And the result is that they obtain a insertion of the human genes into various chromosomes at varying copies. They obtain a group of monkeys, and these monkeys show variable degrees of phenotype. Very characteristically, they show a phenotype of repetitive locomotion, repetitive patterns of locomotion, in this case, a circular motion around the cage. All the transgenic monkeys show much higher, most of them show higher this repetitive stereotype motion locomotion as compared to control. You can do that. They also show higher anxiety, do the standard anxiety test, showed all these transgenic monkeys are more anxious, have higher anxiety. And most importantly, the social interaction of these transgenic monkeys are reduced. Even within the group, compare their interaction with the other monkeys within the group, or in pairs, taking two monkeys, transgenic, or transgenic wild type. They all show very much reduced interaction, especially between pairs of transgenics. So this social interaction deficit is a very useful indication, similar to that of the autistic phenotype. Now, overexpression or duplication or mutation of MECP2 gene is known to cause the Rett syndrome, one of the autistic syndrome or autistic disease. Another case studies to manipulate another gene, which I think is even more interesting. This is a gene controlling the circadian rhythm, one of the core genes controlling the daily cycles of metabolisms of various brain states of the brain. The gene we focus is the BML1 gene. It is known that the BML1 loss is associated with premature aging, and also the BML1, cycling BML1 level is greatly reduced in Parkinson's disease and the Alzheimer's disease. So we knock out this gene by CRISPR-Cas9 technology by the Hongjin Chang's laboratory performed this work by using, by deleting exon 13 of the BML1 gene, produced a number of progenes, 3 complete knockout of the expression of protein expression of BML1, some mosaic expression of the mosaic editing by the CRISPR-Cas9 produced a mosaic reduction, a reduced BML1 level. But the complete knockout is very clear. In all the tissues in the body, the BML1 is absent. Now, these knockout monkeys show very clear phenotype. First of all, the locomotive activity is abnormal. More motions during the daytime or during the nighttime using the periodogram analysis of the motility, and what we expected fits our expectation is the reduced sleep. We look at both the wake state, non-REM sleep, and REM sleep using EEG recording, show that clearly the knockout monkeys show reduced non-REM sleep and REM sleep. And this is even more clear if we do a 3-day light, continuous light illumination. The sleep time is greatly reduced, and both REM and non-REM sleep. So the sleep disorder caused by BML1 knockout has also been shown in the mice. But what's different here with monkey is that there are other phenotypes that are not easily observed in the mouse model. I'll describe that phenotype a bit later. Very strikingly, the very good sign of BML1 knockout is the reduction of the cyclic variations of various hormones in the blood. Melatonin, testosterone, DHEA, and cortisol, the stress hormones, are all flat or reducing amplitude. In particular, the cortisol, the so-called stress hormone, maintain a higher level for both knockout and the partial knockout, the mosaic animals. That suggests the animal is in a higher stress state. In fact, the phenotype clearly show the monkey is highly stressed. They show in their motility, they don't move as much. They tend to hide in the corner, mostly above the ground. This difference between the wild type monkey and the knockout monkey is very clear. Now, all the knockout and partial knockout show clearly reduced, increased stress, reduced motility. Now, we can look at the stress also in the behavior of this monkey in response to people. The caretaker comes over to feed the animal, the knockout monkey hides in the corner, while the normal monkey come and grab the food. Now, the other thing that's absent, phenotype is absent, is that the monkey has EEG patterns that are similar to humans. And they also show this very clear mismatched negativity peaks associated with the arbor effects that are found in the schizophrenia patients compared to normal, this enhanced MMN peak was also shown in our knockout monkey. So there's a phenotype, EEG phenotype. There are also EEG patterns for the sleep disorders. Those are also very similar to the patients with insomnia patients. So all these are indication that these monkeys are good models for studying circadian disorder-related disease. The above two approach, transgenic approach by expression of foreign genes or the human gene in the monkeys, or by gene editing, has problems. The technical problems, the viral introduction of transgenes also mostly random. Inserted genes in various variable copies in different chromosomes. And the CRISPR-Cas9 editing has low efficiency and off-target effects, mosaicism in editing. So there's a difficult, and it's also difficult to do multi-gene manipulations. And more seriously, that we cannot breed monkey within a short period, normally 5 to 6 years. And even though we have various treatment can reduce in 2 years, it's still very slow generation time. But the importance, most importantly, is that there's always a variation in the genetic backgrounds of different monkeys that are produced by these methods. So with this led us a few years ago to the idea that we should clone monkeys to reduce the background variation, genetic background variation. The cloning method is very popular in the past for cloning various mammals, from the dolly, the sheep, many other species has been cloned. And the monkey has been difficult, but we found that this can be achieved a few years ago by producing by specific treatment during the procedure. The procedure involves taking the fibroblasts of the monkey, all uniform fibroblasts. You fuse the fibroblasts with the enucleated oocyte, and the oocyte with the nucleus removed. And the fusion of the fibroblasts with the enucleated oocyte produce that are required for development of the embryo. And after trying many epigenetic modulation, we finally found way that the embryo can develop well. And so that with an in vitro development well developed up to the gastrula stage, we put the embryo into a surrogate mother and produce a clone monkey. So in the late 2017, we produced 2 monkeys named Zhongzhong and Huahua. And we show that genetically their nuclear gene comes from the fibroblasts. Very clear. We can do this short tendon repeats already confirmed, unlike the donor, oocyte donor. But the mitochondria DNA are identical to that oocyte donor because the oocyte contain a large amount of mitochondria that overwhelm the fibroblast mitochondria. All these are what we expected. So this genetically we confirm the nuclear gene is replicated, identical gene are replicated in these 2 animals. And this cloning success of using the somatic cell nuclear transfer have created quite a news in the media. And the Nature had specifically made a comment on this, that this cloning is very useful perhaps for the biomedical research, but it also raised the concern about reproduction cloning in humans. Now, clearly, the purpose of doing this is for the development of animal models for disease. There's no attempt or no intention to do human reproductive cloning. So that worry, even though there are some societal concern, but this is definitely not with the agenda within the scientific community. Now, given the success of these nuclear transfer methods for cloning, we took one of the BML1 knockout monkey, which showed very severe phenotype. This knockout monkeys was done by gene editing of the in vitro fertilized embryo. And their phenotype showed the strongest phenotype in locomotion, in depression, and anxiety phenotype. as well as in their EEG patterns. We took their fibroblasts and cloned 5 monkeys from this. And these 5 monkeys, you can see this table, the efficiency are not very high. For 16 pregnancy we produced, 5 live births was obtained. Now this was out of the 65 surrogate mothers. Now we consider the success for pregnancy in the wild type monkeys. Probably 60%, 70% of pregnancy will result in live birth. Now 60–70% is the goal, that if we can do that with the cloned monkeys, cloned embryos, we would have to elevate our present efficiency 2 or 3 times. But still, this is in the beginning stage of our approach. But eventually, all these 5 monkeys, I should say that we have used this clone relief. Efficacy test, while if the genetic background varies at a great variation. Now, preclinical research needs some sort of animal models, and this animal model should not have a genetic variability, so that we can effectively test the drug efficacy. Right? In the clinical trial, we got to have a large scale, especially in the phase 3 trial, to have large number because of genetic variation. But for effective screening of drug, animal models with uniform genetic background is very useful. Now, this is the donor monkey and the clone that are produced, only 2 months old. They show very similar anxiety phenotype. They hide in the corner. They look distressed. Now, now this one of these monkey, when this monkey was given on the newly developed drug, the symptom was removed immediately. So again, this the knockout of BML1 and the cloning of BML1 knockout monkey was published in 2 papers in National Science Review early last year. And this again created a lot of excitement and debate according to Nature. Now we should talk about the ethical issues of the cutting edge neuroscience. Now there are many aspects of neuroscience that require ethical consideration. Let's start with neuromodulation technology. That's well underway right now in many places. Invasive or non-invasive methods. Now, the monitoring of modulating the individual's brain, that has to be an ethical issue. Drugs that repair or enhance brain functions has ethical issue. Presumably, drugs are used to protect the brain or to cure the disease, but there are drugs being developed that supposedly the cognitive enhancement drug that are under development, even in the industry. So there's an ethical issue about who is able to use this drug and what would be the ethical concern of using this drug in the community. There are genetic technology applied to human disorder. They are concerned over early diagnosis, early genetic diagnosis of brain disorders. Right? This is well-known problems. Do you want to know your gene to tell you that you have certain brain disease? Right? Genetic and stem cell therapy, to what extent one can use this therapy? Certainly, there are already clinical trials on the somatic cell manipulations of genetic manipulation has been approved for clinical trials. And there are also potential stem cell therapies. All these where you can do this, what type of cells you can do, and what are the procedure, what are the ethical criteria that a successful or development of this methodology can be agreed upon before the clinical use. All these are ethical issues needs to be discussed. Certainly, gene editing of human embryo can only be done a very limited extent. For example, 2 weeks in the petri dish. That's the limit one can do. But the frontier research indicate that one can potentially do gene editing that would be tremendously helpful. Right? For example, the editing of mitochondria gene, replacement of mitochondria, that doesn't even need editing. Replacement defective mitochondria gene with a normal one. Is that an ethical approach for mitochondrial disease? All this needs to be put on the agenda for discussion. Now, research using non-human primates is the topic today. I would say that the gene editing or treatment-induced non-human primate model for for the brain disorder are being developed in many places throughout the world. Now, what are the ethical considerations one needs to have for this approach? Now, there are also studies using human cell tissue xenografts in non-human primates. There are production of the human monkey chimera just for the study. Now, human pig chimera has been done. This obviously has this clinical application of producing organs for transplant. All this, whether you can do that in non-human primates, this is, again, an ethical issue. Now, I would say that we need a global consensus for neuroethics. Now, one has to balance the frontier scientific pursuit with societal needs. Frontier science discoveries often push societal advances, but the code of ethics always develop following the discoveries or the development of technology. So once the technology is there, one has to develop codes of ethics. Now, the effective code of conduct could be effectively established. Now we can record the history of the recombinant DNA, which the scientists got together in Osiloma in 1975 to establish a code of conduct for the use of bacteria, genetic recombination of microorganisms. Now, this led to later the NIH guideline, which the widely spread use of recombinant DNA technology throughout the world. And the ethical issues, that code of conduct is established by scientists first and then proposed to the society. And the society accepted and it's been practiced. So this is doable for the non-human primate research as well. There's also the question of universal human values and cultural and religious factors. Now, the code of ethics established by the advanced countries in the West often are biased by their cultural and religious traditions. And it has dominated the ethics, the code of ethics in the past. And there are needs for the developing countries, and there are developments, recent developments of many developing countries. Their voices need to be heard. They should not follow only by the code established unilaterally by the advanced countries in the West. And this is very important in my mind. It should be possible, in fact, to accept a universal code of ethics. For example, we have the 3R principle for animal use. That's widely expressed throughout the world. So we should be able to establish a universal code of ethics through the wide discussions among scientists from all countries. Now, is it possible under this universal code of ethics, a distinct secondary code of ethics? Now, this is balanced by the society special society requirement in different societies. Now, I thought about this with the example of the induced abortion, for example. Induced abortion is not a universal ethic code. And in many states in the United States, based on their mostly religious and cultural backgrounds of the states, have regulations that are totally different. They do not allow induced abortion, right? But in other states they could. So there are secondary code of ethics that's possible, that needs to be discussed. And also there's important for these scientists to really convey the realistic capability of the cutting edge neuroscience without exaggeration. Now the scientist has never thought about cloning the human, and it's not realistic and it's not ethical. And all this needs to be well conveyed to the media, to the society, so that the society have a better understanding of non-human primate research and its benefit. Finally, I would say that this consideration at the national level in the United States has been ongoing. And 2 years ago, the National Academy put in a workshop and published a document. I think this document is very important, very useful. And I also participate in this workshop. It had a very balanced view of the non-human primate research and how one should proceed with further ethical consideration. Finally, I would end with one commentary notes by Andrew Parker, who recently wrote an article about the ethical cost of doing nothing. I'll quote two sentences from the article. He said, "It is essential to be clear that just stopping the research is not a neutral option. Not doing things also has ethical consequences in terms of scientific and biomedical advances that are benefit to the human society. There are also fundamental problems with the precautionary principle of Western policymakers that often adapt to justify their decisions. Now, we should really consider whether these precautionary principles have fully considered the ethical cost of doing nothing." Okay, I'll stop here. Thank you very much.